Hi, my name is Matson Leonard, and it is February 3rd, 2026. Planktonic growth refers to the microorganisms that live as individual, free-floating cells in a liquid environment. Within this state, cells are suspended in fluids such as water, blood, or laboratory growth media. Because they are not attached to the surface or protected by any other cells, planktonic microbes are generally more susceptible to antibiotics, disinfectants, and the host immune system. Although this form most commonly studied in laboratory cultures, it's not really the way that microbes exist in natural environments. In contrast, biofilm is a structured community of microorganisms that is attached to the surface. These cells produce and embed themselves in a self-generated extracellular polymeric substance, or EPS matrix. The matrix is composed of polysaccharides, proteins, and DNA, which allow the microbe to adhere to the surface while also protecting them from environmental stressors. Biofilms can form on a range of surfaces, including living tissue, teeth as dental plaque, medical devices such as catheters, pipes, and virtually any surface that remains moist. Several types of microorganisms are capable of forming biofilms, but bacteria and fungi are the primary formers. Many bacterial species are well known for their ability to form biofilms and are important in both environmental and clinical settings. These include E. coli, Pseudomonas, and Staphylococcus. Fungi, particularly the Candida species, can also form strong and resilient biofilms, especially in healthcare environments. Protists may occasionally be found within the biofilm communities, but the virus is not able to form biofilms on their own. However, viruses can still be present in biofilms by infecting the host cell within the biofilm structure. Biofilm development is influenced by several environmental conditions. Surface availability is important, as rough or irregular surfaces can promote microbial attachment. Nutrient levels are also playing a big role, with lower fluctuating nutrient conditions often favoring biofilm formation. Moisture and moderate fluid flow help deliver nutrients while allowing the biofilm to remain attached. Temperature, pH, and oxygen availability further affect the biofilm growth and stability. Additionally, exposure to antimicrobials or pressure from the host immune system can actually encourage microorganisms to shift from planktonic growth to biofilm formation as a survival strategy. Quorum sensing is the cell-to-cell communication system that's used by many microorganisms to coordinate group behavior based on population density. Microbes produce and release small signaling molecules called autoinducers into their environment. As microbial population increases, the concentration of these signaling molecules increases as well. Once the threshold concentration is reached, specific genes are activated or repressed simultaneously across the population. This allows the microorganisms to behave as a coordinated group rather than individual cells. Many important virulence factors are mediated and controlled by quorum sensing. These include biofilm formation, maturation, toxic production, and the secretion of enzymes such as proteases and lipases. Quorum sensing also regulates the expression of adhesions that help the microbe attach to the host tissues, antibiotic resistance mechanisms such as efflux pumps, and strategies for invading host immune response. By delaying the expression of these virulence factors until a significant population density is reached, pathogens are able to cause more effective infections that are often chronic and difficult to treat.